The immune system is a complex series of pathways meant to protect the body from infection and disease. But what do we do when the immune system starts to react to things we don't want it to, like allergens, transplanted organs, or even itself? In today's video, we will overview different therapies we can use to manipulate the immune system to avoid tissue damage and tissue function disruption. Before we talk about the different methods and mechanisms used to manipulate the immune system, we must first discuss the different ways we classify therapeutic compounds. We can classify therapeutics into two groups based on their molecular weights and synthesis: small molecule compounds and macromolecular compounds. Small molecule drugs, as the name suggests, are low molecular weight compounds that can interact with biological targets. They are typically chemically synthesized but can also be naturally derived. Often they are administered orally, and their small size allows them to be absorbed through the gastrointestinal tract. Their low molecular weight also allows them to penetrate the cell membrane to reach their biological targets. Common small molecule drugs include drug families such as corticosteroids and antibiotics. Conversely, macromolecular compounds are more complex and have higher molecular weight. Macromolecules are often polymeric, which means they are a larger compound composed of smaller linked subunits. Macromolecules are broader in classification and can be broken down even further into subclassification called non-biological and biological, often referred to as biologics. Non-biological macromolecules are synthesized by various techniques, including chemical synthesis or nanotechnology, and include things like polymeric drug conjugates and polymeric nanomedicines. Biologics are macromolecules produced in living cells or organisms, and are composed of things such as antibodies, gene therapies, and recombinant proteins. It is important to also note that often grouped in biologics, though not considered macromolecules, are therapies that utilize cells and organisms, such as vaccines and CAR T cells. Therapies made to manipulate the immune system is an ever-growing field of research with new advancements emerging every year to improve the field. While there are a broad range of therapies, both FDA approved and still in development, we can study them based on their therapeutic goal. Immunosuppressants are treatments meant to decrease the activity of the immune response, while immunotherapies are treatments meant to enhance or modulate the immune system to prevent tissue damage. Both of these methods have their benefits and their detriments, so let's break them down individually. You are probably more familiar with immunosuppressive drugs than you realize, as they are some of the first immune manipulating drugs developed and utilized in many treatments, such as pain and inflammation management, transplant acceptance, and cancer treatment and management. With the goal of decreasing the activity of the immune response, immunosuppressants can do this in various ways. Many immunosuppressants work through systemic immune system suppression, which, while successful in suppressing harmful immune activity, also suppresses beneficial immune activity due to their lack of specificity. Examples of systemic immunosuppressants can be seen in the two small molecule drugs prednisone and cyclophosphamide. Prednisone, which is a small molecule synthetic version of the hormone cortisol, is part of the group of drugs called corticosteroids, which cross the cell membrane and bind to intercellular receptors of the nuclear family, which allows them to exert an effect on the inflammatory mediators, the migratory cells, as well as decrease the number of CD4 T cells. Corticosteroids have beneficial therapeutic effects in part because they significantly increase the concentration of glucocorticoid hormones, exaggerating the body's response to inflammatory mediators. This systemic immunosuppression and exaggerated response is successful in managing pain and inflammation, but can come with a variety of adverse side effects. Cyclophosphamide is a cytotoxic drug which is not only an immunosuppressant but also used as a chemotherapy treatment. This drug crosses the cell membrane and interferes with the synthesis of DNA. They are successful immunosuppressants due to their cytotoxic effect on dividing lymphocytes. While effective, the drug's use is limited because of their effect on all tissues with continuously dividing cells, irregardless of the location of the immune response, causing significant adverse effects such as the hair loss and leukopenia often associated with chemotherapy. Due to the significant detriments of systemic immunosuppression, newer immunosuppressants have been developed to have specificity, targeting specific functions of the immune system and avoiding systemic suppression. Some examples of these targeted immunosuppressants include the biologic alatuzumab and the small molecule drug tofacitinib. Aletuzumab belongs to a class of therapeutics called monoclonal antibodies, which are proteins designed and harvested in labs from organisms or cells that are designed to act like native antibodies. Aletuzumab binds to lymphocytes, specifically the cluster of differentiation 52. When aletuzumab binds to the lymphocytes, it targets them to macrophages or natural killer cells for removal. Monoclonal antibodies are a broad group of therapeutics which contain many different sources, mechanisms of action, and even therapeutic goals. If you want a video about monoclonal antibodies, let us know in the comments. Tofacitinib is a Janus kinase inhibitor, or JAK inhibitor, a class of drugs which inhibit different Janus kinases in the JAK-STAT pathway, which is responsible for cytokine transduction and growth factor signals. The inhibition leads to suppressed inflammatory pathway activation. Both aletuzumab and tofacitinib as targeted therapeutics have less adverse effects when compared to total systemic immunosuppression, but they lack differentiation between healthy and detrimental activity in the areas they target, meaning they still come with their own sets of adverse side effects. Another approach to manipulating the immune system is the use of immunotherapies. While the goal of immunosuppressants was to reduce the activity of the immune system, an immunotherapy's goal is to enhance or change the immune system's activity for treatment. This can be observed in cancer therapeutics in the use of cytokines and checkpoint inhibitors, both biologics which work to interfere and change immune activity without suppression. Cytokines, or proteins secreted by cells, are powerful therapeutics as they can be used to affect the behavior of nearby cells, stimulating or counteracting various immune pathways to provide anti-inflammatory effects. For example, the FDA-approved interleukin-2 is known for its aid in immune regulation and T-cell growth and proliferation, all things important to developing an anti-tumor microenvironment. By delivering the cytokine to a cancer patient, we are able to promote the immune system to follow anti-tumor pathways in a similar way to prednisone by changing the naturally occurring concentration of the cytokine. Checkpoint inhibitors are monoclonal antibodies which work to disrupt the immune system's ability to activate or suppress T cells. In a cancer-free patient, an immune checkpoint works when a protein on the surface of a T cell interacts with a protein on the surface of another cell, which tells it whether to initiate an immune response to remove the cell because it's unfamiliar or sick, or to ignore the healthy cell. In cancer patients, their tumor cells have adapted to have proteins which mimic those of healthy cells, interfering with the T cell's ability to sense and destroy them. Checkpoint inhibitors block the interactions between the T cell and tumor cells, inhibiting the T cell's ability to flag the tumor cell as a healthy cell and allowing the immune system to flag and clear the tumor cell. Two FDA-approved checkpoint inhibitors include ipilimumab, which blocks cytotoxic T lymphocyte-associated antigen 4 from binding to B7 ligands, and pembrolizumab, which blocks cell death protein 1 from binding to programmed cell death ligand 1. While both of these methods are successful in altering the immune system, they still come with their own set of side effects. Both treatments can cause immune system overreaction, which can release a flood of cytokines in something called cytokine release syndrome. In today's video, we briefly covered the classifications of therapeutics, two methods we could use to approach manipulating the immune system, and examples of how different drugs can achieve these approaches. If you would like to hear about any of these topics more in depth, or if there are any other biotopics you want us to cover, let us know in the comments. And if you like this video, subscribe to help our channel grow so we can continue making content just like this.